Armando Hasurungan, biology and medicine videos. Please make sure to subscribe, join the forum group for the latest videos. Please visit Facebook Armando Hasurungan. Make sure to like, and you can also subscribe if you want. You can ask questions, answer questions, and please post some interesting things such as your artworks. And you can also change the quality settings of this video to the highest one for better graphics. And we're continuing on from the membrane proteins part 1. This is membrane proteins part 2. And we're looking at the different types, the different types of proteins and how they are affected by certain chemicals, for example. So here we have an integral protein. And here we have a peripheral protein which faces the cytosol, the inside of the cell. And peripheral proteins, as we mentioned in the first video, can also bind to integral proteins. Now, what causes peripheral proteins to detach, to basically break off the membrane? Well, changes in pH, urea, chelating agents, and also carbonate can cause peripheral proteins to dissociate and basically be released from the membrane lipids. This does not occur for integral proteins. Integral proteins respond to something else. Integral proteins are much more stable because they span the whole membrane, and the hydrophobic region, as shown here, is embedded within the bilayer. So what actually allows integral proteins to be released from the lipid membrane? Well, actually, detergents can do this. What detergents do is that they bind to this hydrophobic domain, which usually is within the bilayer, and it basically surrounds the hydrophobic domain, and this allows the integral proteins to be released from the lipid bilayer. Now, another type of receptor, which I did not mention in the first video, is called the amphitropic protein. And what this protein does is it actually faces the intracellular fluid and responds to a conformational change of some sort and actually has a biological reaction. So it can go back and forth from the membrane protein. And unlike the integral protein and peripheral protein, this does not really require a chemical to be disassociated. The amphitropic protein only requires some form of biological reaction in the cytosol inside the cell for the amphitropic protein itself to be detached off the membrane, and then it can return to its original place after this biological reaction has ceased. The last specific class of protein I'd like to talk about is a glycosyl phosphatidylinositol, abbreviated GPI. And this looks like a pretty complex type of protein because of the carbohydrate chain, the protein, and the lipid, which makes up this type of protein. But essentially, this protein portion of GPI can dissociate by an enzyme phospholipase C. So as you can see, there are different mechanisms for different proteins to dissociate from the membrane lipid: detergents, changes in pH, biological reactions, and enzymes itself. Now let's have a closer look at the GPI membrane protein and also other lipid-linked proteins. So here again we have the cell membrane and the extracellular fluid and the intracellular fluid, and we're looking at the lipid-linked proteins. And we just talked about GPI, which stands for glycosyl phosphatidylinositol. It consists of a lipid component, carbohydrate component, and a protein component. We will look into this GPI protein later on. But there are other types of lipid-linked proteins which do not have carbohydrates. And these vary depending on the fatty acid chain it contains, such as this one, which has a palmitoyl group consisting of 16 carbon fatty acid chain. And as you can see, the amino group and the carboxyl group of the protein is within the intracellular fluid. We can also have a different type of fatty acid chain holding up the protein, such as a fatty acid with a myristoyl group, a carbon with 14 carbon fatty acid. And this will hold a protein typically with a connected to a glycine amino end and the carboxyl end on the opposite side. And so these are lipid-linked proteins, or you can also refer them to as peripheral proteins, which are lipid-linked. Another type of lipid-linked protein is a protein which connects to a phenystyl group. And so these are the variations with these different types of lipid-linked proteins, because they can have all different types of fatty acids that they link to. So we can see some common associations with them. For example, they all contain different types of fatty acids. the proteins connect to different types of fatty acids. And also another important thing is the fatty acid basically holds the protein. Actually, the carbon or the sulfur, in this case, a carbon is actually holding the fatty acid. So one carbon anchors the protein. And so this structure makes it actually very unstable because there's only one carbon holding the whole protein. However, amino acid sequences such as a positive lysine or other positive amino acids can actually attract the carboxyl end of the fatty acid, which will further stabilize this interaction, and so will further stabilize the lipid-linked protein structure. However, lipid-linked proteins are not as stable as the integral proteins, which are the membrane-spanning proteins, if you remember. Now let's get away from these peripheral lipid-linked proteins, but go back to the GPI, the glycosyl phosphatidylinositol. And a good way of remembering GPI, if we must shuffle up the letters, we can spell out pig. And so GPI actually looks like a pig when you think about it. And glycosyl phosphatidylinositol is actually classified as an integral protein, even though it is lipid-linked. So it's an integral protein, and so that's why we see sometimes that there are integral proteins which are lipid-linked, and this is a good example. And why is it lipid-linked? Well, let's have a flashback to the beginning of this video. Remember I mentioned that a change in pH, urea, or addition of chelating agents or carbonate can actually remove peripheral proteins from where they are attached. In this case, a peripheral protein is removed from an integral protein and one that is bound to the membrane. And so these type of chemicals actually release the peripheral proteins. However, carbonate, which are one of these chemicals, does not release GPI. And so that is why it is classified as integral protein, because usually carbonate will work in removing peripheral proteins. Now looking at a common example of a GPI, it actually contains two fatty acid chains which combine with a phosphate and a carbohydrate groups, many carbohydrate groups, and then the protein itself. And we can say that here is the amino end, and that the carboxyl end actually is what anchors, connects with the GPI other structures. And so GPI are actually special because they face often extracellularly, as shown by this diagram. And so what this actually is good for is that it can be used for signaling. And GPI are mostly found in locations called rafts, special domains which are enriched in many types of proteins and lipids, such as they are enriched in glycosphingolipids, they are enriched in cholesterol, these rafts. Rafts are also enriched in certain types of proteins, such as caveolin, and which helps in endocytosis. It is also enriched in saturated fats and also glycosyl phosphatidylinositol, as mentioned. And so all these structures in this so-called raft domain helps in signaling and communication with other cells and also taking in things. Let's look at some other advantages these lipid-linked proteins have. So they have a diverse function, as we've mentioned, for communicating and signaling, for GPI. And these lipid-linked proteins actually have very good mobility, so they're very mobile. They can move around the cell membrane very easily. And a good example of this is GPI, and I'd like to portray this in the small diagram I'm drawing right now. So for example, a typical lipid can move through lateral diffusion, mobility, so move around the same plane at a rate of about 10 times 10 to the negative 8 centimeters squared a second, which is very, very fast. And this is, of course, a normal lipid cell membrane structure. If we look at proteins, on the other hand, an example of this can be peripheral proteins, which is attached to the membrane, or actually an integral protein, which spans the membrane. It moves at roughly about 10 to the negative 12 to 10 to the negative 9 centimeters squared a second. And this is very, very slow compared to a lipid structure. Now, if we look at GPI protein, glycosyl phosphatidylinositol protein, it moves at about 10 to the negative 11 to 3 times 10 to the negative 8 centimeters squared a second. And so these figures, this GPI protein, moves at just a bit slower than the lipid, but a lot faster than normal proteins. And this is, of course, because the protein is attached to a lipid structure and so it can move quite easily. So that concludes the video for part 2. Please like, share, and comment. Thank you for watching.